Okay, last we left off, we were talking about leukocytes, and we're going to begin our discussion with the granulocytes, defined as the white blood cells which, when stained with right stain, will exhibit a certain coloration based on the affinity of the granules for dyes within the right stain, such as methylene blue and eosin. Neutrophils are the most common leukocyte and have cytoplasmic granules that absorb both dyes, and thus it stains their cytoplasm a light pink. These are active phagocytes that ingest and destroy bacteria. They're also known as polymorphonuclear leukocytes. The term polymorphonuclear simply means a cell that has a nucleus that can take on many shapes, right? Remember that words mean things. Poly means many, morpho means shape, nucleo means nucleus. These uniquely shaped nuclei are composed of 3 to 5 lobes. You can see in the diagram at the top what they look like. The injured cells release chemicals that attract neutrophils. These are going to induce a process known as positive chemotaxis, in which neutrophils will exit the blood and release granules in the damaged tissue. The granule contents directly kill bacterial cells and attract more neutrophils and leukocytes to the region and enhance inflammation. Now, inflammation, we're going to find out, is an important process, and when we define inflammation, we're talking about several processes. Okay, number 1, redness at the site, commonly termed erythema. 2, swelling, commonly called edema. 3, pain. And 4, an increase in temperature, sometimes called fever. And all of these processes are designed to bring blood to the area. With the blood come the white blood cells. With the white blood cells come the ability to fight the pathogen and repair the tissue damage. Basophils are the least common leukocyte. You can see the picture down at the bottom here. They have an S-shaped nucleus that appears dark purple as a result of the uptake of the methylene blue in the right stain. And the chemicals in the granules mediate inflammation. So what are those chemicals? Histamine and heparin are among the most abundant. Eosinophils, you can see here, have a bilobed nucleus and appear red due to the granules' affinity for the eosin dye. These are phagocytes that ingest foreign molecules. They respond to infections with parasitic worms, and they mediate allergic reactions by engulfing antigen-antibody complexes. The granules contain enzymes and toxins specific to parasites, as well as chemicals that mediate inflammation. Agranulocytes include lymphocytes and monocytes, both of which lack visible cytoplasmic granules and do contain lysosomes, which are not visible using the right stain. Lymphocytes are the second most common leukocyte in the blood. They contain large spherical nuclei and a light blue rim of cytoplasm. There's 2 basic types. They have similar appearances but different functions, and both types are activated in cell markers found on cells called antigens. B lymphocytes, or B cells, are activated as a result of interaction with a special type of lymphocyte called a T helper cell, okay, or they can be activated as a result of encountering processed antigen on the surface of an infected cell or tissue, and they generate antibodies which bind to and remove antigens from tissues. Each population of B lymphocytes secretes antibodies that bind only one specific unique antigen. An antigen is a collection of molecules on the surface of an infected cell or of a pathogen that is dissimilar to our own, and thus tags that particular pathogen or tissue for destruction by the immune system. T lymphocytes are also activated by specific antigens. They don't produce antibodies. They have membrane-bound receptors for individual antigens. T lymphocytes activate other immune system components and directly destroy abnormal body cells, such as cancer cells or virally infected cells. Antibodies and T cell receptors both bind to only one unique antigen based on their structure, and this is an example of the structure-function core principle. Monocytes are the largest leukocyte. They have a large U-shaped nucleus surrounded by light blue or purple cytoplasm. They only circulate in blood briefly before exiting the capillaries to enter tissues where they mature into macrophages, which are phagocytic cells that ingest dead and dying cells, bacteria, antigens, and cellular debris. They activate other components of the immune system by displaying phagocytized antigens to other leukocytes. Now, complete blood count can be used as a diagnostic tool for the physician to determine what might be making you sick. It's an important test for anemia as well as other conditions. Here are blood samples drawn and examined under a microscope and also analyzed by an automated analyzer to evaluate the number and the characteristics of blood cells. These days a computer can do that. In the old days you used to have to make a blood smear and then manually count the field for the different types of cells that you would see. Red blood cell count are made in cells per milliliter and used to calculate the hematocrit. It's also a direct measure of the hemoglobin concentration. The higher the crit, the higher the hemoglobin concentration, the higher the blood carrying capacity of the blood. The blood sample is also drawn and examined under the microscope and by an automated analyzer to evaluate the number and types of blood cells. The red blood cell characteristics include size, volume, and concentration of hemoglobin in the cytosol, the platelet count and volume, and the type and number of different leukocytes. And then what the physician can do is compare those numbers to ideal numbers and determine whether or not you have a particular disease. Leukopoiesis is a process in bone marrow in which hematopoietic stem cells form new leukocytes. Hematopoietic stem cells divide and produce two cell lines, the myeloid line, which produce most of the foreign elements including the erythrocytes and the platelets, and the lymphoid line, which generates lymphoblasts, which are committed to becoming B and T lymphocytes, which then develop into the prolymphocyte precursor cells. The myeloid stem cell line differentiates into blasts, which are committed to becoming monocytes, called monoblasts, and precursor cells called promonoblasts. Granulocytes are derived from myeloblasts that differentiate into precursor cells called promyelocytes. Band cells or stab cells are the final precursor stage, and they develop into mature granulocytes that enter the bloodstream. B and T lymphocytes mature in different locations. The B in B lymphocyte stands for bone marrow, and the T in T lymphocyte for thymus. B lymphocytes remain in the bone marrow throughout their maturation, while T lymphocytes migrate from the bone marrow to the thymus in the mediastinum and complete their maturation there. Now, again, we're referring, of course, to red bone marrow, right? And in the adult, the long bones of the skeletal system contain mostly adipose tissue, and so with the exception of the proximal heads of the humerus and the femur, most of that marrow is designed for fuel storage. Okay, let's take a listen to leukemia.
Leukemia is a cancer that usually affects the white blood cells. White blood cells develop from stem cells in the bone marrow. Leukemia results when something goes wrong with the process of maturation from stem cell to white blood cell. Usually, there is a rearrangement of pieces of chromosomes that disturb the normal control of cell division. The affected cells multiply without restraint, becoming cancerous and ultimately occupying the bone marrow, replacing the cells that produce normal blood cells. These cancer cells may also invade other organs, including the liver, spleen, lymph nodes, kidneys, and brain. There are four major types of leukemia. Acute lymphocytic leukemia is a life-threatening disease in which the cells that normally develop into lymphocytes become cancerous and rapidly replace normal cells in the bone marrow. It most often affects children between the ages of 3 and 5, but can also affect adolescents and adults. Acute myeloid leukemia is a life-threatening disease in which myelocytes become cancerous and rapidly replace normal cells in the bone marrow. Exposure to large doses of radiation and use of some cancer chemotherapy drugs increase the likelihood of developing acute myeloid leukemia. Chronic lymphocytic leukemia is characterized by a large number of cancerous mature lymphocytes and enlarged lymph nodes. Chronic myelocytic leukemia is a disease in which a cell in the bone marrow becomes cancerous and produces a large number of abnormal granulocytes, a type of white blood cell. This disease may affect people of any age.
Okay, platelets are small cell fragments surrounded by a membrane that are the smallest of the form elements. They're formed by stem cells called megakaryoplasts, and they're involved in hemostasis, which stops hemorrhaging. Platelets don't have nuclei or organelles and are found in most whole cells. They contain granules that have clotting factors, enzymes, mitochondria, and glycogen deposits, and enable them to carry out oxidative catabolism for a limited amount of energy yield. They also contain cytoskeletal fragments that include microtubules associated with actin and myosin filaments. Platelet formation, or thrombopoiesis, begins as hematopoietic stem cells differentiate into committed precursors called megakaryoplasts from the myeloid line. These become megakaryocytes that go through repeated mitosis without cytokinesis, thus generating a massive nucleus with multiple copies of DNA. The mature megakaryocytes are stimulated by thrombopoietins that send cytoplasmic extensions through clefts in the bone marrow sinusoids into the blood, and these break off into platelets. They have a limited lifespan. They last a little over a week, after which they're removed from circulation by the liver and the spleen. Now, hemostasis involves a series of 5 distinct events that form a blood clot that plugs a broken vessel. The primary function is to limit significant blood loss. The first part includes vascular spasm, then platelet plug formation, then coagulation, then clot retraction, and thrombolysis. So let's take a look at hemostasis.
Hi everyone. I want to start off by talking about Mary. Mary likes to play soccer, and she was at one of her games and she was running really fast. She scored a goal, and then she tripped and fell and scraped her knee. And then she started to bleed. Now, normally our bodies want to make sure that our blood, which flows through our blood vessels, stays in our body. But if we have an injury that allows blood to leak out of the blood vessel and out of our body, we want to make sure we can stop that. And we do that through a process called hemostasis. And it's through hemostasis that we create this clot. And this clot allows the bleeding to stop. It stabilizes the injury in the blood vessel, and it gives the blood vessel time to heal. Now let's take a look at exactly what's going on in Mary's knee and exactly how we make this clot. So here we have a blood vessel, and in our blood vessel, we have our blood. In our blood, we have many different types of cells. One of the most important ones is our red blood cell that carries oxygen all over to all our tissues in our body. And in this blood vessel, we make sure that blood is flowing smoothly in one direction. Did you know that if we were to line up all of our blood vessels, the total length of our blood vessels would come out to about 150,000 kilometers, which is about 93,000 miles. This is the same distance as if you were to travel around the Earth a little bit less than 4 times. So you can see we have a lot of work, our blood vessels have a lot of work to do. And one of the cells that helps with this are the cells that line the inner wall of the blood vessel, the cells that communicate with the blood. These cells are called endothelial cells. And it's these cells that allow the blood to flow smoothly, and these are the cells that communicate with the blood whenever there's been an injury and hemostasis needs to begin. Now let's take a look at Mary's knee. She scraped her knee, so let's cause a break in her skin. Let's say this is her skin. And she also caused damage to the blood vessel underneath. So let me erase this. Let me cause some damage to the blood vessel. And so you can see blood that should be flowing in this direction now can come out of the blood vessel and out of the skin in this direction. We want to stop this. And the way we stop this is through hemostasis. There are 2 stages to hemostasis. The first stage is primary hemostasis. And this begins immediately after there's been an injury. And what happens is the endothelial cells at that site of injury begin to secrete proteins. And this sends signals to the blood that there's been an injury. And in our blood we have platelets. These platelets are made in the bone marrow, just like red blood cells, and their purpose is to respond to injured blood vessels. And I drew them as squares, but they're actually about this small compared to a red blood cell. The reason I drew squares is I want to make a note of what they carry inside of them. They carry these sacs or granules that carry molecules and proteins that are necessary for hemostasis. So they store them, and they're ready for use whenever needed. And so what happens is the endothelial cells secrete these proteins and send a signal to the platelets to come to that site of injury. And then a sequence of events happens with the platelets that I won't go into details here, but it allows the platelets to stick to that site of injury. They get activated, they change shape, they release what they have inside, and then they start clumping together and call more platelets and cause more platelets to clump there as well. And what we end up with is what we call a platelet plug. And this is the first step of stopping any bleeding. But this is still kind of weak, and we need to make this stronger. And that's where secondary hemostasis comes into play. In secondary hemostasis, we make this platelet plug stronger with a protein called fibrin. And what fibrin does is it lays over and links up on top of this platelet plug and makes it tighter and sturdier. It's like tying a knot. In order to prevent it from getting loose, you double knot it or triple knot it. And this is exactly what fibrin is doing. But fibrin doesn't float in our blood as fibrin. It carries around an extra piece of protein. And when it has this extra piece of protein, it's called fibrinogen. So we need a way of getting fibrin from fibrinogen. And the way we do that is with this family of proteins called coagulation factors. And I'm drawing them all in one color, but there are several different types of coagulation factors. And the injured blood vessel will send signals to these coagulation factors to get activated. And once one gets activated, it will activate another, and activate another, and activate another. And then ultimately, that extra protein on fibrinogen will get cut off, and that will allow fibrin to link up on the platelet plug and make it stronger. This sequence of activation of coagulation factors is what we call the coagulation cascade. I won't go into the details here either, but it's just key to remember that in secondary hemostasis, we make this platelet plug stronger by getting fibrin from fibrinogen through the coagulation cascade. And it's with this fibrin mesh over the platelet plug that we create this clot. And it's this clot that stops bleeding, allows blood flow to continue through the blood vessel, and allows the blood vessel to heal.
At the site of vessel injury, the first platelets arrive to start sealing the wound. Simultaneously, the coagulation cascade with its various coagulation factors is activated. This involves two pathways, the extrinsic and the intrinsic pathway. Extrinsic activation begins with now exposed molecules of the vessel wall, such as tissue factor, which forms a complex with factor 7. Finally leading to the activation of factor 10. This factor 10A is the point at which the extrinsic and the intrinsic pathways of the coagulation cascade meet. The intrinsic pathway consists of various coagulation factors activating each other in a chain reaction. At its end, a complex with an additional cofactor is formed. This complex now activates factor 10. Since the two pathways merge at the level of factor 10A, this factor has a pivotal role in the coagulation cascade. Further down the cascade, factor 10A in combination with 5A activates thrombin and induces the so-called thrombin burst. One molecule of factor 10A can catalyze the formation of 1,000 molecules of thrombin. These large amounts of thrombin cause the further activation of platelets and the enhanced formation of fibrin. Fibrin then forms strands, making up the mesh that stabilizes the platelet plug in an arterial clot and holds together the red blood cells in a venous clot. It can be concluded that the central role of factor 10A in the coagulation cascade makes it a viable target for therapeutic intervention in pathologically altered blood coagulation.
So what you can see here is the big picture of hemostasis. Again, we begin with vessel damage. Vessel damage results in an initial vascular spasm and an exposure of collagen fibrils. This attracts platelets to the sides of the injury, and they're going to release components such as serotonin that promotes vascular spasm. This will also be released from damaged tissue. Smooth muscle is going to mediate this. In addition, they will release chemicals that attract more platelets. Eventually, you'll see the formation of a platelet plug leading to coagulation. And once the clot forms with the conversion of prothrombin to thrombin, resulting next in the conversion of fibrinogen to fibrin, we get the insoluble clot, followed by clot retraction, and then once the clot has served its role, thrombolysis dissolves the clot and returns us back to the original condition. This is simply a summary of all the components involved in the clotting cascade, and what's important to understand about this is that as a cascade, it represents an effective way of generating a very vigorous biological response to an initially small event. It's produced by positive feedback, another example of the feedback loop core principle. It has to be tightly regulated in order to prevent inappropriate or lack of clotting. Endothelial cells generate two chemicals that regulate the first and second stages of clot formation. Prostacyclin, which is a prostaglandin, inhibits platelet aggregation, and nitric oxide promotes vasodilation. Endothelial cells and hepatocytes produce anticoagulants that inhibit coagulation, such as antithrombin 3, that is a protein that binds and inhibits the activity of factor 10A and thrombin, and so prevents the activation of new thrombin, and heparan sulfate, a polysaccharide that enhances antithrombin activity. Protein C, when activated by protein S, catalyzes reactions that degrade the clotting factors 5A and 8A. Now, there are disorders of clotting that we should be aware of. A clotting disorder is a condition in which clotting is not regulated properly and can create consequences for maintaining homeostasis. The two types are bleeding disorders and hypercoagulation. A bleeding disorder increases hemorrhaging from even minor injuries because the blood can't clot, usually due to a clotting protein deficiency, such as hemophilia A, which is a lack of factor VIII, or B, which is a lack of factor IX. Hypercoagulating conditions result from inappropriate clot formation, known as thrombosis. The clot is dangerous and can obstruct blood flow through vessels. If this happens in the brain, it can cause a stroke. In the lungs, it can cause pulmonary infarction, and in the heart, it can cause a myocardial infarction. Thromboembolism can break off a thrombus and occlude smaller vessels downstream. Hypercoagulated conditions are frequently characterized by thrombi that form in the deep veins of the leg. This is called a DVT, or deep vein thrombosis. The reason for this is venous stasis as a result of the blood having to flow against gravity. A pulmonary embolism is a dangerous complication of a DVT. The emboli can break off thrombi in the legs and lodge in small blood vessels in the lungs. Now, we can use clot busters and anti-clotting medications to fight these conditions. Patients with thrombi or emboli are treated with anticoagulants, which are a wide-ranging group of meds that prevent emboli, including heparin, which is used in hospitals. The action is immediate but has to be injected. Warfarin, which is used for outpatient managing of clots, can be given by mouth. It inhibits the production of vitamin K dependent clotting factors by the liver, and those would be 2, 7, 9, and 12, and does not affect factors already in the plasma and takes about 3 days to be effective. Antiplatelet drugs include aspirin, that inhibits enzymes that contribute to aggregation, and clopidogrel, which blocks platelet receptors and inhibits platelet activation. Glycoprotein 2B and 3A inhibitors are used in heart attack patients in a similar fashion. Thrombolytic agents such as TPA or urokinase restore blood flow rapidly to prevent tissue damage when the thrombi or emboli have caused a stroke or a heart attack. Now, blood transfusions are essentially the removal of blood from a donor and the infusion of blood into a recipient. They are commonly used to treat a lot of conditions. The discovery of surface markers or antigens found on most biological molecules in all cells, including erythrocytes, led to the development of safer transfusion practices. The immune system recognizes foreign antigens and responds by trying to destroy them. The response was responsible for many fatalities in early-era transfusion patients. Antigens on erythrocytes are genetically determined carbohydrate chains that give rise to different blood groups. Erythrocytes are particularly useful for clinical use, and the ones that we pay most close attention to include the ABO blood groups and the RH blood groups. So let's take a look at blood typing.
Type 2 hypersensitivities involve interactions of antibodies and surface antigens of cells, followed by complement-assisted lysis of these cells. A typical example is mismatched blood transfusions. There are 4 different blood groups based on types of antigens on the surfaces of the red blood cells. People who are type A have A antigens, and those who are type B have B antigens on the surfaces of their red blood cells. Persons who are type AB have both A and B antigens, and those who are type O have neither A nor B antigens. The serum of people with type A blood contains antibodies against type B antigens, and the serum of people with type B blood contains antibodies against type A antigens. Type AB serum contains neither antibody, and O serum contains antibodies against both A and B antigens. If blood from a person who is type B is transfused into a person who is type A, antibodies present in the type A blood react with the surface antigens on the incoming red blood cells. This leads to complement fixation and lysis of these cells. If blood from a person who is type A is transfused into a person who is type B, antibodies present in the type B blood react with the surface antigens on the incoming red blood cells. This also leads to complement fixation and cell lysis. Persons who are type O lack A and B antigens on the surfaces of their red blood cells and are therefore universal donors. Persons with type AB blood lack antibodies against A or B antigens and are therefore universal recipients.
Here you can see an example of the results of blood type testing. Agglutination is indicated by the clumping that you see here, 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 and here, whereas lack of agglutination is exhibited here, here, and here. Blood samples from 4 patients are combined with antibodies against each of the antigens in the ABO group. The agglutination indicates that a specific antigen is present in that patient's erythrocytes. Okay, so you can see here in type A blood we have agglutination with the A antigen and not with the B. The inverse is seen with type B blood. With AB blood we have agglutination with both, since both antigens are present. And with type O blood, the O antigen does not have a corresponding antibody, and thus we see no agglutination. Your immune system recognizes antigens on your erythrocytes as self-antigens. It does not produce antibodies to self-antigens. If it did, you would attack your own tissues. And in fact, we'll find out in the chapter on the lymphatic and immune system that this does occasionally happen for a variety of suspected reasons. The immune system does produce antibodies against foreign antigens, and this means that antibodies are present in your plasma only if the antigens are absent from your erythrocytes. Note that anti-A and anti-B antibodies are preformed. They are present in the plasma even if the individual's never been exposed to those antigens, while anti-Rh antibodies are only going to be present in an Rh-negative individual who's been exposed to Rh antigen. As a result, an Rh-negative individual who's never been exposed to the antigen will have no anti-Rh antibodies. Antigens and antibodies are the basis for blood matching. Blood taken from a donor is screened for compatibility prior to its administration to the recipient. A match occurs if the donor blood type is compatible with the recipient. A transfusion reaction is where a recipient has antibodies that bind to the donor antigens, and this causes agglutination that destroys the donor erythrocytes, leading to kidney failure and death. Now you might wonder, in the course of a transfusion reaction, why don't we worry about the donor's antibodies? And it's a good question. And it turns out that, generally speaking, when we take blood from a donor, we remove the plasma and resuspend the erythrocytes in physiological saline, and that dilutes the donor antibody to negligible amounts. It also turns out that even if we were to use whole blood, the antibody present in the donor blood would be in such small quantity that it's not going to produce an appreciable amount of hemolysis. But generally speaking, we give RBCs and saline in a typical transfusion. A condition known as erythroblastosis fetalis occurs when an Rh-negative mother gives birth to her second Rh-positive baby. Okay, and in the old days, we used to have to do blood tests in order to check for Rh compatibility before you got married. During birth, fetal RBCs enter the mother's blood and stimulate her immune system to generate anti-Rh antibodies. The first pregnancy is typically not at risk, but subsequent pregnancies are going to run the risk of the maternal Rh antibodies crossing the placenta and attacking and hemolyzing the fetal red blood cells and potentially killing the baby. It can be prevented with blood type screening. If the woman is Rh negative, she can be given a compound called RhoGAM, which is basically anti-Rh immune globulin. This binds fetal cells in the maternal circulation and prevents the immune system from coming into contact with that antigen, and thus no antibodies are produced. And the result then is she can have as many Rh positive pregnancies as she wants. Prior to that, we had to screen for blood type compatibility, and then if you were incompatible, you had to use judgment in terms of how it is you wanted to approach the prospect of having children. You could adopt, you could get married and not have children, or you might wish to not get married at all in that event. But with the advent of ROGAM, that's no longer a major consideration. So you can see here the 8 major blood types. Now, an important thing to point out when we're talking about blood typing, and we're using terminology here, when we say that somebody is A positive or A negative or O positive or O negative, okay, the positive or negative part refers to the presence or the absence of the Rh factor, okay. This part here, the ABO part, refers to the presence or absence of an ABO antigen. Okay. So the positive and negative have nothing to do with the ABO designation that comes in front of them. Don't get confused by that. So, for instance, if somebody were AB negative, their RBCs would contain the A antigen, the B antigen, and would lack the Rh antigen. If someone were B positive, their red blood cells would contain the B antigen and the Rh antigen. Okay, and you can see, looking at the chart, what the antibodies would be. The antibodies would be directed against anything that's not your own antigens, with the exception here of the Rh antigen, which, if you're Rh negative, you have to be exposed to in order to express in appreciable amounts. Okay, I thank everybody for listening. Remember to review this material, as it will be covered on the PRSs and on the exams, and I'll see everybody in the next podcast.